Hello, everyone. I'm Can. I'm a PhD student in the Safari Research Group, led by Professor Onur Mutlu at ETH Zurich. And as Thomas said, today I'll present our work blend, a fast, memory efficient, and accurate mechanism to find fuzzy seed matches in genome analysis. So in genome analysis, like high-throughput sequencing machines, as we all know, extract smaller fragments of original DNA sequences known as reads. So the challenge is actually to perform genome analysis from the small random pieces of the genomes. So we commonly use these reads to identify genomic sequences or their similarities in two ways. So we can map the reads to a reference genome to basically find the potential matching locations in the genome. And also we can identify the differences between a read and a reference genome. So the second option is to overlap the reads to also each other. So overlapping reads identifies the matching regions between the reads, which then can be actually used to identify the de novo assemblies from these overlaps. So to achieve all of these similarity identifications, we basically must identify these similarities efficiently so that we can facilitate a practical search among many sequence pairs. So, for example, mapping reads to a reference genome usually requires an expensive computation on a large number of reads and a potentially very large space. For example, a human reference genome contains more than 3 billion characters. And so the indexing sequences enables a practical search over a large database of sequences. And hash tables are commonly used for that purpose, like as a data structure. So index can contain some subsequences that we, for example, call chimeras, extracted from the reference genome, and the list of basically locations that they appear in the reference genome. And to identify the similarities, we usually apply several heuristics in several steps. For example, a seeding step can determine the potential matching regions between a read and the reference genome by extracting also some chimeras from a read and then identifying where they appear within the reference genome that we call like candidate regions or the seeds, let's say. And then afterwards, in order to further reduce the search space from the seed matches, we can also apply some seed filtering techniques, such as chaining, for example. Then this will also further identify like highly dissimilar regions in the reference genome so that we can further reduce the search space. And then the last step is usually, and optionally, is to perform sequence alignment, which will basically determine the exact differences and the similarities between a read and the reference genome. So let's look more into the identifying seed matches using hash tables between a pair of sequences. So here we have two sequences. So one sequence can be coming from a reference genome, let's say. The other one can be coming from a read. So what we usually do is that we identify some subsequences from these sequences. Like, for example, we can apply some sketching techniques. But so we're going to call these subsequences seeds, by the way, to let's say, introduce some convention for the rest of the talk. And then to identify basically matches quickly, what we usually do is that we hash these seeds, and then store these hash values in a hash table. And then when we basically want to figure out whether there are matching regions in the second pair of the sequence, we do the same thing. We again identify the seeds, and then we hash them, and then later we query the hash table that we generated previously, and the querying hash table will basically quickly tell us if there is a match basically in the hash table or no match to identify the similarities between sequences. And there are basically like multiple ways of performing seed matching. For example, instead of generating all overlapping chimeras from a reference genome from sequences, we can sample some of these. Like, for example, a very well-known method is called minimizers. So here we have a window of chimeras, and what we can do is that we can hash them, and then instead of picking all of them, we can just pick the one that has the minimum hash value and then store it in a hash table, which basically will significantly reduce the search space to store these seeds in the hash tables without hopefully basically hurting the accuracy too much. And another mechanism is, so we call these, by the way, the minimizer chimeras. So another mechanism is also known as space seeds. So we may want to basically tolerate some mismatches when finding matches between these sequences. And in order to do so, we apply some pattern on that sequence. Like, for example, there are, let me see if I can find, okay, laser. So basically, like, you may want to tolerate some, let's say, mismatches at certain positions. So we may basically introduce some don't care characters at these positions by applying some pattern, let's say. And then we call these seeds space seeds. And then again, similarly, they are hashed and then stored in the hash table. But what we do is that, like, when we find a hash match or a seed match, we find a match on space seeds, although these original sequences may not be matching exactly at certain positions. So the choice of pattern for space seeds is, of course, critical for effectiveness of space seeds. And the last method that I want to mention is, so we may also allow some insertions and deletions between the sequences that we want to identify similarities. For example, hashing this entire long sequence, what we can do is that we can first select some k-mers between these sequences, for example, minimizers, and then we can link them together, which, for example, like, we can call them, as we know, like strobmers from the linked minimizers. And then we can hash these strobmers to identify basically seed matches using these strobmers. So when we identify a seed match, we can identify seed matches, although basically there are, let's say, insertions and deletions between these sequences because we're matching these strobmer seeds using their hash values. But, like, as you probably have seen already, what we're doing, like, essentially we're hashing the seeds to find seed matches with a direct lookup of these hash values. And we usually use low collision hash functions. So this means that the seeds must exactly match, not the original sequences that we, not the original sequences that we basically use to generate these seeds, but the seeds itself need to exactly match to generate the same hash value. And of course, like, this is very advantageous, right? Because then this means that we're not assigning the same hash value for dissimilar seeds. Then it becomes unlikely to find matches between these similar seeds. But there's also one particular limitation is, then this means that we're also not assigning the same hash value, or we cannot find the highly similar seeds that we call fuzzy seeds, using also these low collision hash functions because they are unlikely to match. So let me try to convince you about the challenges of exact matching seeds. So there are main limitations when adjusting the many seeding parameters when we know that the seeds have to exactly match. So one parameter is k-mer size. For example, should the k-mer be long or short? Or, for example, if we're working with the minimizers, should we pick a long, let's say, window or a short window? And then basically all these parameter choices determines the content of the hash table that we use to perform genome analysis, right? And there's a clear trade-off basically when exactly matching the seeds in terms of the performance, memory, and the accuracy. So then let's try to look at the opportunities that we may have when finding the fuzzy seed matches. So a mechanism for finding fuzzy seed matches can enable, of course, assigning the same hash values to highly similar seeds. For example, here the seeds are not exactly matching, but a fuzzy seed matching technique could basically assign the same hash value so that we can find these highly similar seeds by directly looking up their hash values. And of course, we should still be assigning the different hash values for these similar seeds. And such a mechanism also should provide high performance, meaning we don't really want to do a distance calculation or similarity calculation, or it should be also space efficient, meaning like we don't really want to do or use multiple hash functions for a single sketch when performing seed matching. And basically, fuzzy seed matches can enable finding useful and novel seed matches that cannot be identified when finding only exact matching seeds. And also it enables us to, for rethinking the seeding parameters to achieve better trade-off between performance, memory, and accuracy. So let me quickly go over our goal and key ideas in this work. So our goal is to enable finding fuzzy seed matches as well as exact matching seeds with a single lookup of the hash values of these seeds. To this end, we propose blend. Blend is based on two key ideas. Blend uses a hashing mechanism called SimHash that can generate the same hash value for similar sets. And it provides the mechanisms for accurately and efficiently converting seed sequences into a set of items. So let me quickly go over the SimHash technique based on an example to basically generate the same hash values for a similar set of items. So this is an example input that a SimHash can take as a set of items. Here the entire sentence is set, and the items of this set are basically the words in the sentence. SimHash can generate a single hash value for the entire set using the hash values of items. So the hash values can be generated using a low collision hash function. Here we show the hash values in their binary form. So then basically to generate a single hash value, SimHash calculates the net difference of ones and zeros at every bit position and stores this difference result in a counter vector. Here we have the net difference results for each position. Here, for example, the leftmost value in the counter vector shows the difference result for the most significant bit, and the 0 means we have the same number of ones and zeros, and the rightmost value shows the net difference for the least significant bit. So the positive value means that we have more ones than zeros, and also we have like a -2 here, so this means that we have more zeros than ones. So we can use basically we can convert this counter vector to a SimHash value. Then basically, by looking at the SimHash value, we can directly tell whether a majority of bits is 1 or 0 based on the bit that we set at a particular position. So the one important property of SimHash is that when like two sets are generated, or the hash values of when the SimHash value for two sets are generated and these two sets are highly similar to each other, they are likely to generate the same hash value because, so let's change the one word here, it becomes SimHash, their hash value changes, and also the counter vector may change, but it's also likely that the hash value that we're generating won't change eventually because the majority won't change. So the challenge is to do it efficiently and accurately to convert C's to a set of items to use with SimHash. So we do this using Blend. So our goal is to efficiently find fuzzy seed matches with a single lookup. So the input is a fixed length sequence, for example, seed sequence. So we have 3 steps in Blend. So we can first, like, we efficiently and accurately convert C's to a set of items. We have 2 conversion mechanisms, Blend I and Blend S. And then we generate the SimHash value of the seed based on the set of items generated in the first step. And then we efficiently identify fuzzy seed matches by matching the SimHash values of the hash table. So I'll quickly go over the first seed conversion mechanism, Blend I. So we take the sequence and we extract all overlapping k-mers from the sequence. So we call these neighbors, and then we hash them, and then we use these hash values as the set of items in the SimHash. So we also have Blend S, which is very similar to Blend I. The purpose is to also use it for the linked k-mers, for example, stubmers. Rather than using all overlapping k-mers, we use the linked k-mers basically to identify the set items. But then what we do is that when we identify these set items, we can then simply convert this binary to a vector where we have -1 for zeros and +1 for ones. And we do this using the SIMD operations, and then we perform vector summation again using SIMD to calculate the counter vector, and then we decode that counter vector into a hash value. And then we can use this basically hash value with a hash table to identify the fuzzy seed matches. So we are aware that the blend can be integrated to any seeding mechanism, but in our work, we integrated with minimizers. So for the k-mers that we extract from Windows, we use Blend I to generate their hash values, and then we find the minimum from them. Or like we also use stubmers. We calculate their hash values using Blend S. So I'll quickly go over the results in the paper. So we integrated the blend mechanism into Minimap2 to perform end-to-end mapping. We used real and simulated data sets from PacBio, HiFi and CLR reads, OINT and Illumina reads. And we have like human CHM13, HGO2, fruit fly, yeast, and E. coli genomes. We have 2 use cases. First use case is read overlapping. all versus all overlapping. So we evaluated the accuracy, completeness, and contiguity of the DNA assemblies as well generated from overlaps. And then second use case is read mapping to reference genome. So we evaluated the accuracy of mapping and as well as like we performed structural variant calling. So before showing you these results, I want to show you some empirical analysis that we did for fuzzy seed matching. So we calculate the edit distance between collisions using Minimap2 and Blend. So what we see is that there's a nice Gaussian distribution of the collisions over there when using Minimap2 because we have low collision hash functions. But when we use Blend, there is basically a slight change in the distribution towards generating collisions for highly similar seeds. So Blend increases the collision rate for highly similar seeds while keeping collision rate similar to Minimap2 for dissimilar seeds. So very quickly, I'll go over the results for read overlapping and read mapping. So for performance, I'm going to talk about only for hi-fi reads. We have an average speed of 40x compared to Minimap2, and we reduced the memory footprint by 7x. So we can improve the critical parameters without hurting the accuracy, for example, window length and the k-mer. So for assembly, when we generate the assembly, what we find is that we can generate more accurate and complete assemblies than Minimap2 and MHAP, and also better contiguity in most use cases. So fuzzy seed matches can lead to finding novel and useful overlaps. So for read mapping, again, we have an average speed of 1.7x compared to Minimap2 and similar peak memory usage. But what we also observe is that the computational cost of sequence alignment slightly hinders the benefits of fuzzy seed matching. So we also perform structural variant calling using Sniffles2 and TrueVary. And what we find is that we also Blend leads to generating best overall accuracy in downstream analysis. So this is basically tier 1 high confidence sets from genome in a bottle data set. So to conclude, finding exact matching seeds introduced limitations in further improving the performance and accuracy of genome analysis. Our goal is to enable finding the fuzzy seed matches as well as the exact matching seeds accurately and efficiently. We do this by employing SimHash and then identifying the sets for SimHash efficiently. Blend provides significant speedups and also lower memory footprint, especially when using hi-fi reads, and also improves the accuracy of important applications in genome analysis. So Blend is published in Nucleic Acid Research, Genomics and Bioinformatics. We also have our source code. So with that, I'll conclude my talk. Thanks for listening.